Hi, good evening, everyone. Thanks for the organizer to invited me to give a talk on this. My talk will be a little different than GM. Actually, we're working with plant-based vaccine technology suited for rapid response, and I will give example about influenza. Just outline for the presentation, I will speak a little bit about background of Fraunhofer US Center for Molecular Biotechnology and plant-based system for biopharmaceutical production, mucosal and parenteral delivery, transgenic and transblastomic plant, and transient expression system. Case study, and this is really I'll show in the end of my talk, about the influenza expression, how long it take compared with the egg expression or production of influenza. This is just about background about Fraunhofer company. Fraunhofer is German company actually, or German institutes, and they have 56 institutes in Germany. One of them which work with biotechnology, the rest of them was like nanotechnology and laser, and if you remember MB3, they invented this technology. Fraunhofer in USA, we have like 7 centers. One of them is Fraunhofer Center for Molecular Biotechnology, and our work is concentrated in vaccine production in plant. Actually, most of our influenza vaccine or seasonal or pandemic, which is produced in egg, and if I talk about the production of vaccine in eggs, it takes like roughly like 1 year from inoculation to harvest, purification, and deliver. Egg has like a system, but still have lots of disadvantage. One of them, some people are allergic to eggs, and in eggs, we remember for Chiron, like 2005, 2006, we have contamination, then they have to throw the vaccine out. We need something in to have something really fast to protect the population, as it happened in 2009 with H1N1. There is a multiple system for vaccine production, from bacteria to insect cell, egg, and every system has advantage and disadvantage, but never plant produced vaccine goes to the market. So plant-based expression system started, I guess, 1990 with transgenic plant, but transgenic plant has long lead time, low yield, non-uniform expression, gene silencing. Field production will be like have to go through the regulation and regulatory issue and commercialization question. In 95, plant to go over the lower level of expression, people try to use like plant virus as a vector. It's a little genetically stable vector, non-uniform expression again, silencing, and you have to do transcript to infect your plant. Transcript, in a lab, in vitro transcript is really costly, and if you use inoculated material to re-inoculate, then the viruses start to recombine and they give you like virus infection. And then lately in 2000, there's agroinfiltration, which has become as a method to deliver DNA to the cells with a binary vector. Again, we go back to the same question: a very low expression level and unstable, transformation. So what Fraunhofer tried to do, to combine what is the problem, why every company tried to produce vaccine in plant fail in some stages. So we come with to adapt the system and automate the system to produce vaccine and just try to compete with industrial production. So the idea of Fraunhofer actually, if you don't move from academia to industrial, there is a huge gap between academia and industrial. So someone has to connect academia with the industrial production. First of all, there is a word that's generated by Charles Ernsten. He said, you need to produce edible vaccine. And this is a great idea to have, banana with, some vaccines or tomato or whatever you think about. But the question is, when you come to the FDA, they will never allow you to eat tomato with vaccine, because first of all, they said how you sell it. Did you put them in a pharmacy or you put them in a market? How you say this banana was vaccines, this banana was not vaccines. And the other thing is how much dose you give. Banana is not the same size. Tomato is not the same size. How much vaccine you have in this tomato. So we'll never allow to have edible vaccine. So Charles Ernsten regret his word, after like few years. So I will go over this one and look for how Fraunhofer tried to produce vaccine in plant. First of all, you have three things you have to combine, which is a vector used to deliver your vaccine or gene of interest, the vacuum infiltration, how to scale up your vacuum infiltration. Third, hydroponic system, because if you said, okay, I'm going to grow the plant in field, then you come to the regulation. So, in field, you cannot control the environment, nutrient, whatever you add to your plant to grow. So what we did, actually, we start with the vector, how we combine viral vector with binary vector to have a lunch vector. So we take, if you look here, we have the viral vector, which has replicase, movement protein, take off CP, which is, if you have the CP from the target, then you can have recombined virus and it comes to be the virus again. So we delete, CP, and we use the CP promoter to express your protein. Then your protein under two promoter from movement protein and promoter from CP. And we use this virus and insert it in a binary vector, which is under 35S promoter and NOS terminator with left and right border. Okay? So this is the first thing we did. We tried to improve our vector. How this works? When you add, when you transform your agrobacterium, this is transient system. This is not stable transformation. So when you deliver, when you have your agrobacterium harboring your lunch vector with a gene of interest, for example, like malaria, or I don't know what I'm doing here, okay, with the target gene, what happened is agrobacterium phenomena that they have, very gene which, cut the border, left and right border, and cell attach, bacteria attach it to the plant cell and try to firing single-stranded DNA. Single-stranded DNA goes to nucleus, transported to nucleus, and 35S, you have the first transcript. First transcript in nucleus move to the cytoplasm, right? And then the virus replicate in a cytoplasm. So you have machinery of replication, going on in your cytoplasm all the time. You have RNA replicate, you have translation at the same time. So your level of expression move from milligram to grams of proteins. Okay, and I will show how you look with GFP as example. So you have two systems in one vector. So first you have to choose your agrobacterium cell, system and transported to the cells. And this is natural phenomena. And then you have your fermentation. Why we go for fermentation, not the flask? Because if you go for industrial, you have to grow the bacteria under, GMP condition. The people will not accept any vaccine which is, I don't know, it's like producing R&D. It has to go through current GMP, good manufacture, production. Then you have to optimize your plant, your system, which is, if we try to use soil, but soil is not controlled to grow plant. Still you need to synchronize your plant when they grow. And you have to tell that how much nutrient they add. You cannot tell the FDA from batch to batch there are some differences. They will not accept that. What we came up with, we try to compare different agriculture media. We look for wastes with like sponge. We look for rock wool, which is another media for growth. And as you can see, rock wool is the best media. They have zero nutrient. They have zero nutrient. You add water with the seed, Bentimiana seed. This is 6 weeks old Bentimiana, so they didn't grow at all. There is no support. Then you control your media all the time. You use Hogland, you use hydrosol, whatever you use, and this will be like continuous. There is no change. Like you cannot use city water until regulation. I'm using city water. City water change every time. So that's what we come up. DI water, and you add your nutrients. So you know exactly how much you add every time, and you synchronize your growth. The other question is come to how to use a small food cubit to grow as much as you can, from your plant. Because you cannot grow them outside in a field. Everything has to be contained in one place. We come up with this cascading system, which you grow your plant in tray, and you have, light, it's calculated how much difference between plant and plant, how much difference between, plant and light, and this is all calculated. And cascading, which is you have, vertical, you can grow at like, some people have like 15 level high. Okay, this is, okay, I'll just put them all in one. So how this works, actually, you grow your plants cascading in rock wool, as you can see, to like 5 weeks, and then you grow bacteria, either in flask, you inoculate your fermenter, and then you come to vacuum infiltration. You need to scale up, you cannot go infiltration by hand, plant by plant. So what we did, we generate like, vacuum chamber, which is capable to infiltrate like kilograms of material at once. So you put your bacteria in a tank, you flip your plant upside down in the agrobacterium. It's actually agrobacterium diluted to 0.1, which is very low concentration. You don't need too much. And I'll tell why we have to have them very low. And you apply vacuum, what happens when you apply vacuum? You try to de-aerate the intercellular space from the air. So all the air in mesophyll will come out when you apply vacuum. When you release your vacuum, the bacteria will be infused to the intercellular space, or the stomata, and attach it to the cell and start firing your single-stranded DNA. This process will take like 2 minutes, the whole process. And after that, you leave your plant for 4 to 7 dBi, and then you harvest your protein. So your expression will be from 3 to 7 days, no longer than 7 dBi. I forgot to mention that the vector, Lynch vector, usually has silent suppressor, so you don't have to add silent suppressor. If you do infiltration with binary vector, you still have to add silent suppressor. Otherwise, your protein will be declined within 3 dBi. All goes down to 0. So it's actually it's enforced marriage between bacteria and plant. I don't know what's happening here. I guess this is a movie. I don't know if this will work or not to show you the infiltration. I have to sit on. No. Let me see. It's not playing. That's okay. It's not playing.
So your flash, the video in the...
You want to see it?
Yeah.
I put it in. That's okay.
Okay, no problem.
No problem. But it will be good to see it. Let me have it, You can click this one, I guess, yeah. See if it works or not. You don't have this. Okay. Enjoy the video for like a couple of minutes. Show you how you have how they do the infiltration. There's no voice. It's just showing how we do the infiltration. She's presenting, this tank, it's supposed to have the bacteria. There is water here, just to show how the infiltration works. And you can add like 1 kilogram at the time. And when you apply the vacuum, actually, it goes to about like 28 or 15 parts per million. So now you see the deaeration of the air from the intercellular space. And when you break the vacuum, plant try to breathe again, they have nothing around the leaf except the solution. So with the bacteria, so you infuse it in. And then most of the leaf will have bacteria and ready for expression. As you can see, most like 100% of the leaf infiltrated now. So. Oh, wait. I just need to close this. I need it again. Okay, and this is just an example. I will have another one later, please. Yeah. Thank you. So this is just an example for one tray, as you see, infiltrated with agrobacterium harboring lynch vector. And here in your left hand, this is uninfiltrated material with GFP. Here is infiltrated material with GFP after 7 days. So it's actually you're covering all your tissue expressing the protein or the vaccines. And if you have some part which is not infiltrated, the virus already have capability to move from cell to cell. They will move and produce your protein also. So I'll just give an example for how much we produce. We use in our system Nicotiana bentimiana, 1 gram seeds, 1 gram of the seeds will give you like 10,000 seeds. So it's a lot of seeds. The 1 gram seeds is enough to grow 50 kg of plant material within 5 weeks. So, biomass, it's not a big problem. But now is a problem, how you seed and you move it for large scale production or GMP production. So we try to build the facility. Then we have the lynch vector, which is improved. We synchronize our growth of bentimiana. And we have, generate high cell density of agrobacterium in a small volume of fermentation. You don't need too much from agrobacterium, but you still have to grow him under GMP condition. So we try to optimize the condition to get up to 25 OD of agrobacterium. And then we build, this is all custom made design because it's not available in post infiltration where you keep your infiltrated material for like a week, and then you go for harvest clarification. Here is your agrobacterium goes one way, and you clarify your extraction here, and then you go for purification and fill and finish in your after purification in filling suite. Rooms will be like different class. It's not all the rooms the same class. This room is unclassified, this is unclassified, this room classified 100,000, this classified as a 10,000 room. Again. I'll show you the movie from how the process, the flow of the process. This one. Here's this one. Okay. It's not working. Working? Okay. That's fine. So as you can see, this is a maquette, as I said before, a layout. And he shows this rooms. Your pre-infiltration. Your tray, first go to the seeder. This is automated seeder, no one touch it. You put the seed in a seeder and go 1 seeds per well in a tray. You have 200 spots here in a tray. Seeder will be by vacuum, take 10 by 10 and put them in the right place. After you put them in a seeder, you put them for growth. This will take like 5 weeks in a rack. And then after they grow, the robot will take it. Everything is automated. No one touch it here. Goes to the infiltration, through the window. Air comes from pre-infiltration to infiltration. You flip an infiltration room. This is by a flipper. And after you flip, you move into the post infiltration. From post infiltration, go to the harvest in a different suite. It will show up. Here is the harvest. This is also automated. No one touch it. You just flip your tray, and they have a scissor here, cutting plant. So the shoot to the dicer, which they cut plant to very small small pieces. And then go to the homogenizer, which homogenate the tissue to a fine solution or slurry. And then they move to the tank. You move them to the tank to purification. This is a centrifuge. You try to take all the debris out, and you have only the solution coming out for purification suite. Okay. And you have the filter, 0.2. So if you have any bacteria or any other stuff will stuck in the filter, filtrate, goes through the wall to the bags. And bags move to the purification suite. We have a person actually pushing the bag to the suite, The whole part before we see, this is automatic. No one touch it. You just, have employee watching the process. There's too many rooms because you have to gown and degown. It's actually different than, R&D. Here is your purification skid, iMac or HEC column or Q Capta or Capta Q. And then finally you ultra purified and come and you put your protein for filling in bottles, and you take it to the storage in the same room. As I said, this room is classified 10,000, must be very clean rooms. So, thank you. I'm not late. Oh. Okay, this is just to summarize what we did. Actually, we go from greenhouse growth, bacterial growth in a flask. People has to infiltrate manually, and you see JFB grown where you infiltrate, and then you go for purification. This will not go for industrial. This is R&D, it's okay. But for industrial, no one will accept it. Then we have to move to the medium scale. You cannot move from bench to industrial right away. The people will not accept it also. So we try to make medium scales, which is between industrial and R&D, which as I show, how you grow bacteria in Rockwell, move it to fermentation, vacuum infiltration, and harvest and purification Dyson. This mimicking exactly what happened in a GMP facility, but with GMP facility will be with a large scale. And now we said, okay, yes, we can produce vaccine in plant in industrial scale. Okay, because this goes to the clinical trial phase 1 and phase 2. Doesn't show up. I have a very nice figure. Okay, swine flu in 2009. Then try to see the system. We said, as this is from Washington Post, I didn't make it. So this one, I like it, so I put it here when we have the swine flu. Then we have, okay, let's try to test. Did we tell exactly what we're going to get, or we're just saying, okay, our system is better than egg-based vaccine, or we, hiding something here and there? No, okay. In 2009, everyone remember the break of H1N1, swine flu, and swine flu, there is two protein, which is hemagglutinin and neuraminidase, and everyone believes this is a immunogenic gene in the virus. I'm not talking about the virus too much. And if you have shifting or drifting, you will have different, infection. And people were really afraid, the influenza move from swine flu to human and transmitted from human to human. It will be a disaster like 1918. So let's see how fast we can produce vaccine to protect as much as we could, not everyone, with, faster than egg or any other system. So we're given the gene, say day 1, which is influenza from California. We received the sequence day 1, okay? For 7 dBi, we've done cloning. In 1 week, we have cloning. Plant was growth already in 4 weeks. And we have the target in agrobacterium transformed. And in day 14, we infiltrate our plant. In day 21, we have the protein out. So it's 21 days you have subunit vaccine available to protect people. Again, if you compare it with egg, it's really, really fast. So if you have vaccine within 21 days, you have to, but again, you have to produce it in an industrial scale, not in lab scale. So I know this guy. I've seen him before. So how many, target we try to process the system? We have influenza. Actually, the system is suitable for developing country. It's very good for developing country. For one reason, capital cost is very low. Yes, we pay more for this facility, about like 25, 30 million, and 30 million for industrial vaccine production is nothing, is neglected. You're talking about if you have fermentation process, you're talking about 500 million to $1 billion to build a facility, which is sometimes will be too much for developing country to spend this money to produce one vaccine. The good thing of our plant system, actually, you can switch from target to target without a problem. For fermentation, you have to prove that your fermenter is clean for second product. You cannot go from vaccine to another vaccine in the same fermenter without cleaning. The fermenter. For plant, it's different. The other advantage is if you have fermentation, you have more chance for contamination. And you throw out 10,000 liters of fermenter, see how much it costs, a lot. The plant work as, each individual plant, working as bioreactor by itself. They didn't contaminate each other. So this is also advantage for our plant system. We try to convince the people this is the best anyway. So, we did a lot of work, especially with neglected diseases in Africa, like plague, RSV, malaria, sleeping sickness, trypanosoma, yellow fever from Brazil, and human papilloma virus. And we did some work with monoclonal antibody and anthrax, and growth hormone also for human. So, there is multiple of subunit vaccine which is produced in plant as well as therapeutic protein antibodies. This is all the expressed material in the system from influenza, pandemic, and seasonal. I will not go over all this list, just list. And then, again, when you have you have to, again, as I said, if FDA will not allow you to have, fresh or edible vaccine, then you have to go through purification process. Purification is very simple: harvest, or by clipping the blade, chop the biomass, homogenize in extraction buffer, clarify through, Kono or point to filtration. Target purification, you use IMAC, which is a chromatography column. And you can also, go further purification with ultrafiltration or using anion exchange, purification column. So, purification is available. It's material which is in the market. You can choose whatever you want. Let's give you an example for the influenza, because you cannot move, again, you cannot move from bench to industrial. You have to have, intermediate. And all the assays you use, it has to come up with the same result. If you have different between, medium scale versus R&D versus production, then it will be not accepted. So I'm showing here how we purified from 1 kg material. And you can see this is influenza subunit vaccine here with Kumasi, with Western also. You confirm it, and then when you move to 5 kg with the toxic study, you have the same result, the purity, and if you look for, 50 kg, it will be the similar exactly. If you see here another band, for example, then this stops the process right away. You have something, sorry, I'll move faster. Okay. And you check actually with reverse, with the UBLC, and as you can see, there is no difference between 1 kg, 5 kg, and 50 kg. Again, you have to tell FDA, because in plant system you have to have signal peptide as well as if you target it to ER or cytoplasm or nucleus, whatever you target, you have to tell him where your protein in N-terminus start. Otherwise, you have something, if you have extra amino acid, then it will be rejected right away. So what we did, we sequenced the N-terminus, and we confirmed this is the first nucleotide. First amino acid, second amino acid, third amino acid. So all the signal peptide already cleaved, and you start from amino acid number 1. Safety, I will skip this one. I will go through the immunogenicity of the H1N1, and this is the experiment which we designed to check the immunogenicity, bleed before and pre-bleed. First administration at 0 day, and second administration at 21 day, and then you have pre-bleed and 21 day, 31 and 42. And this is how much we inject. HAI, we call them HAI, and this is intermuscular, and you have 60 microgram, 30 and 10, and the same, but you have adjuvant to alhydrogel. And this is your control, which is, saline solution. And you look for the result for hemagglutinating inhibition, and you see 30 microgram, 60, 30, 10. There is no inhibition. When you add alhydrogel with 60, 30, or 10, you still have inhibition, agglutination inhibition. And then we move to the virus neutralization. As you can see, again, I will not go for this one, doesn't show any virus neutralization. When you have alhydrogel, you start seeing, virus neutralizing, result. Then you have to go through the rabbit tox study, because you have to show that there is no side effect from your vaccine. So you have to go through a tox study. Usually they did it in rabbit. And the same as, like 2 administration doses, 3 weeks, and you bleed during the 3 days. I will go through this very fast. And as you can see here, serum also showed that, there is inhibition in these 2. This is the average of 8, I guess 10 rabbits. And if it's above 40, we consider you have the adjuvant is administered. Okay, and then toxicology study through the rabbit also. You have 10 rabbits, and you have HAI 45, 90, 90 microgram of HAI, and this is the day of injection, day 0, day 14, and boost in day 28, and you sacrifice 5 animal at day 30 and 5 animal at day 42. And you look for the side effect. If you have any side, you look for clinical. If they have any inflammation where you injected, you have to look for health of the rabbit. They lose weight, they eat normal, they didn't eat normal, and if you claim that there is no effect, that will be accepted. And you go through the pathology, and you look for hematology and chemistry, and as you see the result, no gross lesion or noted. So everything is fine from this one, and this is the result of the toxicology study, again, similar to what I showed before. And we went to phase 1 clinical trial for 3 different targets so far. And that just acknowledge for all Fraunhofer R&D group, DARPA, which is supports this project, was building the CGMP facility, for Bill and Melinda Gates, because they give, excuse me, they support, all the African diseases like sleeping sickness, malaria, and stuff. So we're still working on. We have malaria in clinical trial, but it's a different, vaccine, which is called, transmitted blocking vaccine. And thank you.
Save questions until the end, because we do have one final speaker in this section, and we'll please don't run away. We will have a question and answer session at the end. So to finish up our third presentation in this session, novel bacteria-mediated soybean immature seed transformation with Dr. Kate.
Thank you. First I'd like to thank the organizer giving me an opportunity to talk about our effort in transforming the soybean development method. And as you know, the soybean is a very important crop, and not only for basic research, but for our commercial product development. And demand for soybean has been increasing quite significantly over the last decade or so because of the human population growth. And efficient soybean transformation is very important for biotechnology and for the trade development. So soybean transformation, the method is not new. It's actually one of the crop that has been transformed 20 years ago. And but the main problem is at that time, the method developed in 1988, it's called a seedling coordinating node method, and it was patented by Monsanto. So for us to develop a product, so actually we need to develop an alternative method. That's one of the driver. And also there's a method for particle bombardment developed more or less at the same time. Even though it's developed, neither of those methods are very efficient at that time. And also there's a particle bombardment of embryogenic cell cultures. So also if you look at all the, just the transformation development, the technology development, so starting with the cotton method, people and even Monsanto, they went on to develop a more efficient embryonic axis, embryo tip method, and using organogenesis. And then the other company developed a leaf-based method, or actually we developed some of the leaf-based method too. And also I think BASF has developed some leaf node-based method. And there's also work on using other target tissue immature cotyledon, and also organogenic callus. And also using, that's mainly using for agrobacterial-mediated transformation. For bombardment, people mostly using a callus-based transformation method. So there are also just the general for transformation development. So there are a lot of treatments that people have done that can increase the transformation efficiency, including use of antioxidants. That can be not only for soybean, it can be also used for quite a few other crops I know of, including monocots. And other issue we need to solve for product development is not only model lines, you also need to solve the elite line transformation. You need to develop method there, so your product development cycle is much shorter. And also you can improve the agrobacterial strains, and also selectable marker to make it to have a better marker. Not only using like antibiotic resistant genes, but also you can use like a better herbicide marker or non-herbicide marker like PMI and sugar-based selection. And the other things people have been always trying is a new target tissue, try different target tissue to develop, so you can find a good target tissue. So in order to develop a very efficient system, and so on and so forth. And also the last one is particularly for industrial use is automation is very important. I think back in 2005, I think Monsanto they put a lot of effort in there, and they have a very efficient, I think, automation system for not only for soybean, also for cotton, and but for that, was still, I mean, a lot of a challenge compared to even using all those technology, right? Compared to many other field crops like the maize and the rice. And the efficiency, I mean, the basically number of transgenic plants each person can produce, it's still lower compared to those crops. So improvement is needed for in all aspects of the transformation process to meet the increased need of the research. So if you look at the like efficiency, right? It's like a soybean compared to maize. It's about like half or 1/3, depending on what kind of method you use. And soybean is more effort intensive because there are a lot of tissue culture, like chopping, moving tissue, that kind of effort. So to improve that, so we're just looking at the whole process, starting from gene delivery method and to the tissue culture regeneration systems and target tissue and the sellable marker optimization. So we look at through all of that, and we identify some of the areas, like for delivery, for agrobacterium strain, you can improve the strengths. Back in the 2000, people using different strains, right? Starting from like C58 based all the way to the Cry5 based strains, and also rhizobium strains. People have done a lot of improvement. And also the target tissue, it's not only cotton node. People tried like embryo axis and hypercaudal, leaf node, immature inflorescence, and organogenic callus I just mentioned before, even implanter. So we finally focus on, we try to focus effort on the immature, like seeds, because that's something new. It gives some potential. And also for the markers, people have used it before in a lot of like MPT2, the antibiotic resistant marker, hygromycin, and also a lot of like others like herbicide genes like EPSPS, bar gene, PET genes, AOS, and also we have new like HPBD and DMO, all those markers. So finally, we look at all those methods. We decide to use immature methods. So if you look at soybean seed development, and we finally pick the stage from like R5 to R7. So mostly around like 6 and 7 before it's mature, before it's yellowing. So we use that target tissue, and also we compared the method using embryogenic method, because embryogenic method potentially could be very high throughput because you are using callus as a tissue. So we did that, evaluate that too, and so we were able to develop some method to increase the efficiency of using embryogenic callus agrobacterium mediated transformation. But so that's some of the efficiency. So using embryogenesis, even though it's working, but the efficiency is not very high. So we decided to give up on that and focus on the immature seeds. So for the immature seeds, after like some effort on optimization, we were able to develop quite a reproducible system using agrobacterium infection of the immature seeds target tissue. So actually, this is the target tissue. Let me see. There's no pointer. So anyway, it's the tissue with after infection, it goes through tissue culture regeneration. We can obtain the transgenic plants in about 3 or 4 months, and so the efficient transformation frequency, this is using the bar gene. Back in the time, we were kind of collaborating with Bayer on developing HPBD herbicide tolerance trait. So those are the first few experiments. We were able to obtain quite an efficient transformation frequency. Even here, it's quite variable because it was we started beginning. So so you can see actually the method can be applied to different varieties, including like a model variety, Williams 82 Jack, and also some one of our Syngentas quite an elite variety. So the efficiency it's pretty good, and also you can see the we also use the glyphosate selection, and it works quite well, and anywhere from like 3 to 10, I think, and later in kind of production work, we're able to achieve about 8 to 9% transformation frequency. Yeah. So another improvement we made is, as mentioned like this morning at the talk, and in the legumes in general, there's some problem in the rooting, especially like in elite soybean line. So we find out that if you transplant the shoots, if you take care of the shoots directly to soil without any in vitro rooting, you can increase the rooting efficiency greatly. So by using direct transplanting soil, the survival rate it's pretty high. I think the average is about 90%. So it varies from 85 to 100, depending on the shoots that you send to the soil. And this method, direct transplanting, can be applied to all the elite lines, and we have tested quite a few elite soybean lines, and it worked very well. Actually, for the soybean like elite lines, if you root it in agar, usually the leaf drops off, you lose the transgenic plant. That increased our recovery quite significantly. And I think this could be used for other legumes too, I think. And so this is all the survival rate, and also the last column is just like an average transformation frequency. So you can see anywhere from like 5 to up to like 15%. And so in summary, we have been quite successful in using several alternative targets to generate transgenic soybean plants. And we have developed a novel transformation system based on the organogenesis of mature seed tissue. It's suitable for commercial production. So the method has been used in several elite lines transformation, and it's compatible with different selection agents in different, including quite a few different herbicides, including like glufosinate, glyphosate, misotrione, hydroxyzidine, and of course it's compatible with antibiotics too if you want to use it. So it has been used, the technology has been used for generating large number of transgenic plants for commercial trait development, including like herbicide tolerance and insect resistance. I think with that, I think it's, that's all the content I want to present. And at last, I want to acknowledge the people, including some of my former colleagues that's in bracket. I think that's all. If I'd like to answer questions if you have any. Yeah, thank you very much.
So we have some time for questions. So any questions to the three presentations? Yes, please.
Jose Georges from NRC Canada. My question is to Mark Albertson. I'm just curious as to which phytates did you choose to overexpress in sorghum?
You mean the lightweight secondary?
No, the phytates.
The actual phytate. Oh.
Yeah.
You know, right now I'm drawing a blank on that. I have to look at my notes on that. I believe it was just one from.
Is it?
I just don't remember. I'll look it up for you. I'll let you know.
Okay. The other part of the question is, I'm sure you know that there are other, there's a lot of methods to reduce phytic acid. I'm surprised that you gave up so easily with maybe just one trial.
Well, and the reason we did, and we only gave up down regulating phytic acid because of the germination thing. And so that's why we thought we have to do it very differently. What we want to do is keep the phytic acid around as long as it can be, but only get rid of it during the cooking process. And so that's what we gave up on is getting rid of it throughout the life of the plant. So we want to keep it there so it does all the other things it's supposed to do. But it was that germination thing that really kind of freaked us out,
Right, yeah.
because we just couldn't mess with that. So that's what, like I said, that's when we had to go with doing it at the back end rather than doing it throughout the life of the plant.
Okay.
If that makes sense, but that was our thinking anyway.
We have a paper that's in press that in which we are reducing phytic acid production in canola, and it's using metabolic diversion, and the target there was not to reduce it completely to 100% or 80, even 80 or 90%, but to achieve an acceptable level of phytic acid for both the plant usage as well as the industrial purposes.
Yeah, and what we found was that we didn't have to reduce it very much for zinc. So if we were only going after zinc, we would probably still be going down that path. But it was with the iron that we had the problem, because it looked like we had to reduce iron, or we had to reduce phytic acid about 80% for us really to have the impact on iron bioavailability that we ran. And that was the issue that we had.
Thank you.
Another question here, please. Yes. Yeah.
Margaret.
Doctor.
My question is to Dr. Manan, please. Dr. Manan Shamroon.
Yes, here, in front.
Yes.
Okay. Thank you. I have two, maybe you can say simple or trivial questions. The first one is the biosafety in your bioplant with regard to the workers. What level was it?
Or worker? Yeah.
Your definition the staff. What are the containment or the whatever the biosafety level? Labs,
As I mentioned in my talk, every room has a safety level. Like if you are in pre-infiltration, not too much safety. You're free, you grow out. When you move to the agro-infiltration, no one goes to the agro room during the infiltration time. So you don't need to be safe because you'll be outside. The only safety you have to really be in what they call PPE when you go for the purification suite, which you have to be at very high level. That's why you see like a couple of places which you gown and be-gown and all the stuff. So that's what
the other question is compared with yours most sophisticated private plant for production, how do you compare it with the animal vaccine production?
edible vaccine. As I said, Richard Ernst regret this word edible vaccine because it cannot be done. You cannot give tomato to someone, say this tomato is influenza vaccine. It will not be accepted. First of all, it has to, the FDA requested to be processed, somehow. You cannot put it as a tomato or as a banana or whatever. Yeah, I know it's a raw material, good stability for your protein. You have protein in the native form, it's not purified. It will go through the gastrointestinal delivery. It's all safe, right? And easy and cheap. But FDA will not allow under any circumstances except if you purify your protein. And they ask you a clear question: how you control the dose? If you give it like fresh material or raw material. You cannot say, okay, this tomato have 10 microgram or 100 microgram. Otherwise, you have to have some way to convince them that there is no difference between 50 and 1 gram, and they will not accept that. They really respect that, but you cannot say how much will you apply was pre-IND. Pre-IND for phase 1. You're talking about 500 pages to convince him that the vaccine is safe, and you show him the data from toxicity in rat and ferrets and animals. There is no inflammation when you inject it. There is no lose weight. There is no change in liver function or spleen function and so on. So there is a lot of information.
But still, it is cheap and available.
Yes, I will say.
For rural countries.
Yes, I will say.
Rural cities and rural places.
Yeah.
When you can't reach people with vaccine, you can't sometimes. This every vaccine is actually for the poor areas where you can't reach people with the vaccine.
I cannot differentiate people.
You are in the business.
I understand. For me, if you have vaccine, emergency vaccine, which is you have Influenza as H1N1, and which can like millions in a few days.
It is in banana and
yeah, that's what I'm saying. It's better to save the people, than to let him die. Okay, maybe this vaccine will be good or bad. I will use it and save at least 50% from the 50 people. But FDA will, that's the regulation we have not, and you cannot say, okay, I will give it to the poor people, and it will be like it will come with word discrimination right away. So why you give it to the poor people? Yes, our word, the system for one is very cheap to be in a development country. Because with 50 million or 100 million, you can have facility which produce vaccine, and it can be, you can change your target every time. It's very easy to clean. It's very simple because you don't have contained, tank which, you don't have fermenter. You produce your protein in. Your protein produced in leaves, not in a fermenter. So cleaning, changing the target is very easy. And I represent building a new unit. The same, we have the technology, they're building a unit for European. This plant-based system. I hope they will be successful.
So, just a minute. So, Dr. Abmenam, this is, we are using here the plants just for producing the raw material of the needed chemicals, This means that I believe that it's not difficult to understand that it cannot use banana for controlling disease. However, you can still using many plants with the active ingredients as a raw material. And this is the industry, the pharmaceutical industry is based all on this, So you're getting the raw material, trying to extract and purify, and you go ahead with this. How much manpower involved in this kind of unit you showed us here?
Oh, this is very few people. This facility has like 6 employees. They're just 6 employees. They're just watching from outside what's happening. The only room you need 2, when you put your tray, you just put in a tray and robot will take it automatically and move it automatically. Sealer is automatic. You don't have to touch the seal. The sealer was designed to have vacuum. So the banana seed is very, very small. Barely you can see by us, but the needles can make only one by vacuum and put it in the right place. And the other place you need, which is the only room you need 2, the infiltration room, which grows that bacterial cleans the ferment. And then the same people, when you harvest, you will be in a when you purify, you will be in a purification seed. And you don't need really highly, qualified people, just like a social system, when you train them, they will do it. Because it's a process, you have to be SOPs that goes like 1, 2, 3, 4, to avoid any deviation. Because you cannot go to FDA with multiple deviations each batch. It has to be reproducible. Every batch should be the same production.
Okay, thank you. Please.
Okay, Thank you. Margaret Karembo from Kenya. My question was partly addressed by my neighbor here. It is about using edible material, edible fruits and plants for vaccines. And I think this is one area where the application of the technology for the poor would actually backfire badly. On the contrary, because most of the times, most of these, I mean, it will be very difficult, first and foremost, to control what people are going to eat, because in times of scarcity, it will not matter whether the fruit or the crop is a carrier of the vaccine, it will be eaten by many. But now, in the area of the plant-based vaccines, now, what are your sources of the starter plants, and how do you ensure that if at all you get a commercial product, that the benefits are going to be shared equitably from the communities where you get these plants?
It was the same point, actually. I don't know how to answer this, because this is like two conflicts between edible and parental vaccines. We still have, we have to use raw materials, which is easy, as I said, to deliver your vaccine. So, but first of all, before you deliver the edible vaccine, you have to be sure that it's immunogenic, right? Number one. Second, if it goes through the gastrointestinal, how you convince that it's really protecting so the intestine? Still a question mark, this really work 100%. It could be the same between person and person or different. I cannot tell, right? The absorbance of the so, but when you inject material through the blood, it is going through the blood. When you purify protein, and it's immunogenic in animals, immunogenic in ferret, immunogenic in rabbits, there is no toxicity there. Cost, I'll tell you, whatever assessment plant is less than $1 per dose. Less than $1 per dose, which is very cheap. It's a cent. It's a few cents per dose. Because if you produce like 10, like 2 grams of vaccine in 1 kilogram of plant material, that's what we reach. Then, and your administer is like 50 micrograms. Then your cost will be like 50 cents or more. That's if you don't use your column, you regenerate. The most cost in plant system is a column when you purify, right? Because ground Quintiliana is not. You just grow seeds. Shoving is just a sheep, really fast. The most cost when you purify. And if you regenerate the column like 10 times, but you have to validate the column to convince also the regulator that I clean it, I use it again and again and again. Then you reduce your cost to nothing.
So we move to another question, please. Yeah, please, you.
At the back. The other way.
Yeah.
Again, a question to Dr. Monim regarding the purification process. So are you using the HVAC purification or HISTAR purification system, or both of them?
We're using HISTAR purification. There's a HISTAR with subject vaccine.
Okay. Another question, what the percentage of the target protein and the total protein?
HISTAR?
The percentage of...
Yeah, that's a good question, actually. This is when you're using your R&D. Most likely, HISTAR is very stable with protein. It's not cleaved off. If you put it in terminals, then you'll have a problem. I didn't see like cleavage of HISTAR, except that there is like donor and receptor in the DNA sequence. And this is easy by, computer program. They will tell you if you have the cleavage site or not. And if you don't have this, you will have your hashtag on the back. But there's still some competition with, hashtag, which is, tag protein come from the plant. Some of them has like 2 histidine. Sometimes they bind to the column. That's why you move to the second column, like, Q column or any ion exchange column. So further verification. Because they will not accept less than 95% for clinical trial. So you have to put like 2 columns at least.
Okay. Yes, please.
Yes, morning. When I say may I ask you as well? Have your company tried vaccinia as a...
No, only
why not? You can have multiple vaccines by vaccinia.
We use
Okay, try it, okay?
Okay. Do we have? Yes,
Can I
please.
just make a quick comment? Now, from you have said that you have abandoned. Have you tried using edible vaccines and abandoned the process? Now, so this is actually not possible.
No,
Nobody
it's
is
not
trying
possible.
to do that
No.
because it's a major communication issue. Because this is something that really scares people when they imagine that very soon we are going to have crops in the supermarkets that are actually vaccines. And so even as you try to encourage the use of the technology, when people imagine what scientists are doing, putting viruses in plants so that they become vaccines, it gets very difficult
yeah, that's a good
to
question.
communicate.
Before you talk about
It's like determination technology.
First of all, this is the MD virus, which is a plant virus, and it's not the full gene. It's only like movement and replicase. So you don't have virus particles in your production, okay? Yeah, but you don't have really virus in your production. But second, before you have your batch, you have to do virus clearance. No matter what you do it in egg, you do it in pig, you do it in this one, you have to have the result, which is called virus clearance, before you submit it to clinical trial. So you have to have this assessed.
Yeah.
What is
Okay. So, yeah.
Thank you. The same speaker and the same presenter. I'm not sure which diseases you are targeting. But if you tell me the diseases you are targeting, I want to let you know that in South Africa, there is a large consortium that are working on actually edible vaccines for TB, HIV, and even malaria. But what they came up with was that fruit or whatever plant will be given a different coloration that it will be available in the supermarkets. So it will be only be available in certain outlets that can monitor the usage and the application. But I don't know which diseases you are looking at.
For Africa, we work with sleeping sickness, trips. We're working with transmission blocking vaccine, not vaccine, but transmission blocking vaccine, which you block one of the malaria stage or oocyst, okay, to be like, not fertilized, okay, they cannot become the oocyst to move to the human. So, but it's not vaccine to the human, we just protect it in a different way. And we're working with neglected disease like hookworm, which no one cares about. Well, there's too many people, big companies like Novartis, whatever, they will never put that. They will never do it. Like, I know we have here in Egypt, like hepatitis C. No one will do too much work in hepatitis C. For multiple reasons. They mutated, regularly in a patient. I'm not talking about population. I'm talking they mutated in the same patient. So how do you do vaccine for this? So the people doesn't touch it. They work in academia, but they didn't touch it as a production. Company will never work for that. So it will not touch it. This we can touch it here because it's very easy to use the system and again, like hepatitis C again, how many types you have? You have 6 different types. Which vaccine have to go for Egypt, for America, for South Africa, for different places. So there's so many questions. If you don't do your work by yourself in developing country, no one will do it for you. Okay. So we have to go here. Somehow we have to learn, do it, we have to reduce it. No one will solve our problems.
Okay, please. The floor is yours.
I have a question concerning to the development of vaccine in
We have 2 other speakers in this group that maybe we should get some questions for the other speakers. Well, because all of the questions are going to him, right? That's not fair to the other speakers. That's not fair to him either.
You can switch to others if anybody interested.
Yeah, just say, are there questions for the other speakers?
And the other one is
let me answer this one first because I will forget the third one. First of all, the advantage of plant that they're capable of post-modification. If you have legislation, legislation. The only thing is you have in plant xylose and fecals, right? Human being has minerals, right? So you have a tool. You should have transgenic plant which gives you minerals or more than fecals and xylose. So this is called the tools for the system.
But the structure and the building of the glycosylation in mammals is different from.
That's what I'm saying.
So
your engineer, there's engineered in Tignana, which they give you, they mimic the minerals in glycosylation similar to the human. It is. So you can control and sometimes you don't need the exhalation because if you look for malaria, this parasite doesn't have the exhalation. So you have to have a system to de-glycolate also, not just glycolate. Like what do? You do engineering plant with DNJs inside. So the plant produces DNJs when your protein express, they do the glycan.
The small point remaining from the first question is concerning to you have a tag of your protein, and this tag, if it still exists with your protein, it will be immunogenic, and this immunogenic peptide might stimulate the unknown immune system that might attack the human cells and develop the autoimmune disease.
It's known that his tag is immunogenic. I have no doubt about that. But when you have your target protein, because you have like 3 phases, phase 1, they allow you to use a his tag. Second, you have to cleave your his tag, or you have it without tag and purified with amino acid chain. So it's there.
I have just a comment to Dr. Mena. I disagree with you when you said that the virus will not transformation will not form virus particles because you are using transgenic tobacco with cod protein. It has to move.
No, there's no cod protein.
There's no cod protein. It just moves without cod protein.
No cod protein. No cod protein in the protein.
And how the virus is going to move without virus particles?
Cell movement.
Already? Doesn't need the virus particles? Because I don't need the virus.
So plasmid is not the RNA can, most of the plasmid is not the
RNA.
I didn't show that focal microscopy. You have you concentrate it, and you see only the epidermal cell expressing your protein. And if you cell from the mesophyll, when you have the virus moving from cell to cell, you can see your protein in all type of cells, even companion cell, companion tube. Only cell, not cell, but zyme, you cannot see your protein because it's not
systemic. From cell to cell, but not systemic.
It's not systemic, it's cell to cell.
So this system is a pattern?
So with this, I think we concluded this session because we're already 1 hour behind the schedule. And I want to thank the 3 presenters for the very nice and excellent presentation they presented here. I want to thank the audience, and I want to thank my co-chairs. Okay, thank you very much.